Good morning, all of you. In last lecture, I will discuss the microbial assay. Today we will see the next part of our syllabus, that is same the microbial assay. So what is microbial assay? We will see the microbial assay, then advantages of microbial assay, then disadvantages, and the assay of amino acid. So what is microbiological assay? The microbiological assay is defined as the qualitative and quantitative determination of it is defined as the qualitative and quantitative determination of any chemical compound from a simple or even complex material with the use of microorganisms. Yes, I am repeating the definition of this. The microbiology is defined as what? It is qualitative and quantitative determination of what? Any chemical compound from a simple or even complex material with the use of microorganisms. The antimicrobial agents are assayed for following reason. First reason, to determine the potency of the agents, then to determine the pharmacokinetics parameters of drug in an animal or a man, then third, for monitoring and controlling the antimicrobial chemotherapy. For monitoring and the controlling antimicrobial chemotherapy. Then the many therapeutic agents which inhibit the growth of microorganisms. The many therapeutic agents which inhibit the growth of microorganisms or the antibiotic or are essential for their growth, that is vitamins and amino acids, it can be standardized by microbiological assays. Then advantages of microbiological assays over the chemical assay. First advantage over the chemical assay, that is first, it can be performed for the qualitative and the quantitative determination of antibiotics. So it means the first advantage, that is it can be performed for qualitative as well as quantitative determination of antibiotics. Then it is accurate as a chemical method. Then it is simple, then it is specific, then it is inexpensive, then convenient method in order to determine the concentration as well as the potency of the sample. Then disadvantages, have a minimal requirement of space, then as well as have minimal requirement of labor, then materials and time. Then however, technical expertise is essential. For this method, the technical expertise is essential. Then second disadvantage, more difficult to perform than the chemical assay. Then it requires proper calibration and are less reproducible. It requires the proper calibration and are less reproducible and have a greater errors as compared to chemical assay. Then it is, if a good alternate physical or chemical method is available, then microbiological assay are not used. Then it is useful in the quantification of drugs, then vaccines, toxin. Then it is useful in quantification of drugs, vaccines, toxins, then quantification of poisons, then quantification of disinfectant and antiseptics. Then these are useful to determine the specificity of the compound. Then analysis of complex compound like vitamin B12, it can be done effectively by bioassay. Then samples with different chemical compositions have same biological activity. And then last, useful for the sample where other method of assay are not available. I am repeating last disadvantage. Useful for sample where the other methods of assay are not available. We'll see here the chart which shows the name of antibiotic and the name of test organism. For the assay of amikacin, the Staphylococcus aureus is used as a test organism. Then for amphotericin B, Saccharomyces cerevisiae is used as test organism. Then for bacitracin, Micrococcus luteus is used as test organism. Then for bleomycin, Mycobacterium semigatis. Then for carbonicillin, Pseudomonas aeruginosa. Then for deoxycycline, Staphylococcus aureus. Then for the assay of erythromycin, the Micrococcus luteus is used as a test organism. Then for Pharmacetin, then Bacillus pumilus is used as a test organism. Then for gentamicin, Staphylococcus epidermidis. Then for kanamycin sulfate, Bacillus pumilus. Then for kanamycin B, Bacillus subtilis. Then for the assay of neomycin, Staphylococcus epidermidis. Then for the assay of novobiocin, Staphylococcus epidermis is used as a test organism. Then for the assay of nystatin, Saccharomyces cerevisiae is used as a test organism. Then oxytetracycline. Then for the assay of oxytetracycline, Bacillus aureus or the Staphylococcus aureus. Then for the polymyxin B, then for rifampicin, Bacillus subtilis is used as a test organism. Then for tetracycline, Bacillus aureus are used as a test organism. Next, we will see the what is turbidimetry assay for antibiotic, and write the advantages of the turbidimetry method over the diffusion assay. First, they depend upon the growth of microbial culture in a uniform solution of the antibiotic in a fluid medium that is favorable to its rapid growth in the absence of antibiotic. The advantages: the incubation period required for the growth of test organism is short. The advantage is that its incubation period. Then disadvantages over cup method. First disadvantage: this assay is affected by the presence of solvent residue or other inhibitory substances. Then a standard curve is plotted by using a 5 different concentration of the standard solution prepared by dilution of the stock solution. Then next, a medium concentration is selected and the test sample of the antibiotic solution is adjusted by dilution to obtain approximately this concentration. Then 1 ml of each concentration of the standard and the sample solution are placed in each of the tubes. To each tube, 9 ml of nutrient medium previously seeded with the test organism is added, and all the sets are prepared in duplicates. Then 3 control tubes are prepared simultaneously. Culture control, blank and medium control. Culture control, blank and medium control. In culture control, tube containing inoculated culture medium. Then in blank, it is prepared like a culture control to which 0.5 ml of dilute formaldehyde. Blank, it is prepared like a culture control to which 0.5 ml of dilute formaldehyde solution is added immediately. And then for medium control, uninoculated culture control, all the tubes are placed in incubator at the specified temperature depending upon the test organism for 4 to 5 hours. After incubation, 0.5 microml of dilute formaldehyde solution is added to each test tube to stop the growth, and then the turbidity is developed due to the growth of the test organism, and which is measured by determining the absorbance at 530 nanometer of each solution in the tubes. Each solution in the tubes against the blank. And for diffusion method, as I discussed in previous lecture, what is diffusion method. Then advantages over diffusion method: it is rapid and more sensitive method. Then it is simple to perform than the diffusion assay. Then The diffusion assay are more complex. The chances of getting no inhibition zone is higher if the test organism is killed during the spreading. If the test organism is killed during the spreading or the pouring, no growth will occur on the plate. Then, hence, no zone will be observed. Then, next, the dose response in terms of inhibition of the exhibition zone to the concentration of the antibiotic is linear over a small range of concentration. Then, chances of microorganism getting killed during inoculation is very rare during the turbidimetry assay. Then, it can be performed in test tube and also in micro titer plates. Then we will see next, that is the principle and method of assay of amino acid. The turbidimetry method for the assay of amino acid, it is precise, then it is rapid, then it is adaptable to a large number of samples, and the enzymatic method cannot ideal with large number of samples, and the titrometric method is requires a long culture time, that is 72 hours. I am repeating, the turbidimetric methods for assay of amino acids, it is precise, rapid and adaptable to large number of samples. Then enzymatic method, it cannot ideal with large number of samples, and the titrometric method, it requires a long culture time, that is 72 hours. Then test organism L. arabinosus. This procedure used for turbidimetric assay of L-glutamic acid is based on the titrometric procedure and conducted with necessary modification. The stock culture of the organism is maintained in a culture medium by weekly transfer and maintained in the refrigerator. For this, Baker yeast extract 10 ml, then glucose 1 gram, sodium acetate 1 gram, polypeptone 1 gram, and agar is in powder form, it is 1 gram. Then medium for maintaining the L. arabinosus 17 to 5 cultures. Another medium was used to grow the culture for the assay and having the time 7 to 18 hours at 37 degrees Celsius. Then Baker yeast extract, it is 100 ml, then glucose 1 gram, sodium acetate 1 gram, polypeptide 1 gram, salt A 2.5 ml, salt B 0.5 ml, and quinine 0.1 gram. The main culture was carried out in 2 ml of liquid medium used for the assay, and it is also mentioned in above discussion of the above table. Glycine 0.1 gram, D-L-alanine 0.2 gram, L-valine 0.1 gram, L-leucine 0.1 gram, then L-isoleucine 0.1 gram, DL-serine 0.1 gram, then next L-threonine 0.1 gram, then L-cysteine, it is 0.1 gram, same here L-methionine 0.1 gram, DL-asparagine 0.4 gram, L-proline 0.1 gram, L-phenylalanine 0.1 gram, and L-tyrosine, it is also 0.1 gram. Here we can see the quantities of the same, the tryptophan, lysine, arginine, histidine, all those. Then after the 16 to 18 hours of the incubation, the reaction was stopped by the heating at 100 degrees Celsius for 10 minutes. Then each tube was diluted by the addition of 2 ml of water with hand shaking. Then the turbidity was measured by spectrophotometer. Then value obtained by the use of distilled water was taken as a control. Then standard concentration response curve were prepared each time, and the amino acid content in samples was calculated by interpolation. Then method for determination of serum amino acid. For that, example, streptococcus fecalis. Then for this maintenance medium, lactobacilli agar at 4 degrees Celsius for 24 hours. Culture in 10 ml lactobacilli broth, centrifuged and deposit resuspended in 20 ml of sterile saline of a suspension, that is 0.5 ml, and made up to the 20 ml by the use of your sterile saline. Then appropriate bacto amino acid medium is used for actual assay. Then L form of all three amino acids. Then the L form of L amino acid are used 100 milligram L-valine and L-leucine and 50 milligram of L-methionine dissolved in 100 ml of quantities of distilled water of analytical grade purity with your as quantities are double. Then working solution prepared by diluting the 1.0 ml, that is 1 ml of above solution to 100 ml, that is valine and leucine, and 200 ml of methionine with distilled water. This gives a final concentration of, that is 10 microgram per ml valine and the leucine, and the 2.5 microgram per ml methionine. Then the glassware used is Pyrex test tube measuring 115 to 16 mm fitted with aluminum caps. Chemically cleaned and sterile glassware are used. Then sera were obtained from healthy volunteers, otherwise healthy patient undergoing minor surgery. And samples collected by venipuncture before subjects received their mid meal. Then sera is stored at 20 and deproteinization was done by the precipitation with the acetic acid. Then for that assay procedure, 8 tubes were used for the assay of 1 to 8 containing standard amino acid solution, that is 0.0 to 2 ml, that is 0.0, 0.2, 0.4, 0.6, 0.8, 1.2, 1.6 and 2 ml. Then water is added to each this test tube to make a total volume, that is 2 ml, 2, 1.8, 1.6, 1.4, 1.2, 0.8, 0.4 and 800 ml. 2 ml double strength assay medium is added to every tube and microgram tube of amino acid added. Then valine and say leucine, it is 0, 2, 4, 6, 8, 12, 16 and 20. Then for methionine 0, 0.5, 10, 0, 0.5, 1, 1.5, 2, 3, 4, 5. All tubes are set up in a triplicate. After autoclaving at 10 abs for 10 minutes, each tube is inoculated with a appropriate test organism, and all are then incubated at 37 degrees Celsius for 17 hours. After this shaking of the resulting growth and the uninoculated blank, the tube 1 as a no amino acid in this tube and no growth, and are measured against the water in a optica CF, it is spectrophotometer. Then optical density is OD of each triplicate set of tubes. After subtracting the blank is plotted against the amino acid content of the tube, and the amino acid content from the sample is extrapolated on the standard graph.